So hello everyone, as you can see, we have the topic of today's seminar is about data security and data management. I myself, Edward, Robert, and Suresh are going to take a charge of this Saturday seminars, sharing our experience of working with data over years. And we want you guys to learn a little bit about what's going on. Our new era is the era of data. We are generating data every day from our smartphones to work that we do, to even single question like, hey Siri, and the response that we get is just a few kilobytes of data that we are generating. So very simple. It's very important for us to learn more about data. I don't know why I can't. Okay, so the outline of today is I'll be talking about PHI, HIPAA, and IRB. After me, Edward will take a charge to talk about IP in the laboratory, good record keeping practice, integrity in data collection. Then Robert will talk about all kinds of securities. And then Suresh will join us and talk about data quality. So what is PHI? PHI stands for Protected Health Information. For any organization that work with any kind of human data, PHI comes to picture. So it's under the US law. So it's not only some term, but it is under the US law. It's any information about the health status of any individual, provision of healthcare, or even payment of the healthcare that is created or collected by a covered entity. So by covered entity is any business that works with health information. And any information. Okay. And any information that can be linked to specific individuals. So this is interpreted. Okay, so can you mute yourself first? Thank you.
David, can you mute Jacob Hound?
Okay, I resume. So any information about the health status, provision of healthcare, or the payment of the healthcare that is created and covered by a collected covered entity. That means any business that work with the health information that can link to a specific individual, that is interpreted rather broadly and include any parts of patient's medical records or payment history. Now, why it is so important? Leak of any protected information about any patient that can cause to specific patient to be known by public, it's basically leak of PHI. And organization over past decades has been sued for leak of PHI information for millions of dollars. From Los Angeles hospitals to universities, when nurses, they just knew specific actor or actors coming in, they look at their file. That means the PHI data been leaked and they've been sued for millions of dollars. So now you understand why it is called protected health information, because the health information about the patient can be easily identified. So now let's look at what are these identifiers. Any of these identifiers on its own is considered PHI. Now forget that if we have 2 or 3 of this information together, then it's leak of a lot of information. But any of these 18 identifiers considered as PHI. I quickly go through them. The name of the person, by name I mean first name, last name, or even the middle name, or even the abbreviation of the names, dates, except year, telephone numbers, geographic data, fax numbers, social security, email address, medical record numbers, account numbers, health plan numbers. Actually, that's health plan account numbers are together, sorry. Certified or licensed numbers, vehicle identifiers, device identifiers, so URLs, internet protocol, IP address, biometric identifiers, including fingerprints, full face photography, any of these will consider as PHI data. So it's a list of them for you guys to look what considered as PHI data on its own. Okay, so let's see what we have next. The next thing is HIPAA. So let's see what HIPAA stands for. HIPAA is the Health Insurance Portability and Accountability Act of 1996. So as you can see, it's something that I think 104 United States Congress has signed, and President Bill Clinton signed it by 1956 to modernize and protect the healthcare information. Stipulate how personally identify information maintained by the healthcare. So you might be wondering, okay, we are not working in the healthcare, but I need to add any organization that they do research on human, either collecting data from their IQs or their behavior to the sample. It's also considered as personally identifiable information. So don't get misunderstood by the word healthcare and say that, oh, that's not applied to us. I'll go a little bit in details later on of why it is basically something that we need into consideration working with the human data. It prohibits healthcare, again, like any research organization that does human studies, provides and the healthcare business from disclosing protected information. Only patients and patients' authorized representative with their consent can access protected information. You must have heard from your parents that you go to a doctor and then you change your maybe doctor, but then you need to have a consent to release your information. That's how the law is about. It's a little frustrating. It's my information. I want it. But no, sorry, you have to write a consent. Even your parents can sign on behalf of you to be your representative. They have to sign a consent to access protected health information. It can't be even just a tooth decay, okay? Which we consider it as not very important information, but under the law, it's a protected health information about any patients. So let's see what we have next. So HIPAA compliance rule, that means we're talking about the privacy rule, we're talking about the security rule, enforcement rule, unique identifier rule, and code set rule. So first of all, they have to make sure that the privacy of every patient is safe. And then they have to make sure that the information that we provided to them is secure. And then we have to make sure that anyone who works with this kind of information is already aware how to handle the PHI data and how the enforcement come to the picture. Now, okay, so these are this many information. So you go to doctor, you have, you provide your name, your last name, your insurance ID, they have your health record. So what now? Or you participate in a research study. They ask you to give your blood sample, they ask you to give you saliva sample, they run some behavioral data, they ask you to fill up some questionnaires, they have all this information. Now what to do? So they generate a number that is a random number generated by a computer. Now all the data is basically under that unique identifier code for every individual. So now instead of having the name of you when you're doing research study or looking at your files, they look at a number, which is basically, it's a code. The rule is that this code has to generate in a very random way that will not be able to recognize. It needs to be saved in a secure, basically database that maps that code or that identifier with the PHI data. So basically the research, the researchers that they work with your data, they work with that unique identifier code rather than with the PHI. And same thing with the healthcare system. So anytime the words PHI come to picture, it's accompanied by HIPAA, of course. So protected health information or PHI is often mentioned in relation to HIPAA and healthcare, but it is considered protected health information under HIPAA. So these two kind of always come hand in hand together next to each other. So now you might come across different terms that sounds similar, but let's see what are the other ones. So that we want to talk about the difference between PII, PHI and IIHI. So PII is personally identifiable information that's been used outside the healthcare. So what does that mean? You go to, you want to get an app, install an app. So what is happening? So you're entering your name, your age, your email address, your phone number, your gender. For example, it's just, maybe a workout app. You will enter your height, your weight, your BMI. So basically you're providing a lot of information, which is considered as personally identified information for you in the app. So that app is storing all this PHI. So what is it considered? For them, because it is outside the healthcare context, it is considered as PII. But the moment that it's associated with your health information and the healthcare, or I would say the covered entity, universities and research organization, the moment that it comes for them to take a use of it, it's called protected health information. Now we have IIHI, individually identifiable health information. Again, it's same as HIPAA, but again, it's going to be used for outside a healthcare context. PHI is used hand in hand with HIPAA, and PHI and IIHI are both protected by the privacy and security. Now, so they don't have to be compliance with HIPAA when it comes to PII, but the organization have to make sure that all this personal identifiable information is protected under the secure rules and databases. So let's see what we have next. So now we have all this information. I think I give you a little bit of information about the identifier, the identification code. But what they do, they use the de-identification method, is the process to use to prevent someone's personal identity from being revealed. Data produced during human subject research must be de-identified to preserve the privacy of research participants. The biological data may be de-identified in order to comply with HIPAA regulation. Common strategies including deleting or masking personal identifier. So there are so many organizations that if they don't have a secure database to save the information, what they do, once they collect the data, they assign it to a code, and then they'll delete all the personal information. If they're secure, basically they'll map it, but they keep it in a very secure database. Now here is the thing. I'll give you, I give my personal experience. So when I do neuroimaging, when we collect fMRI data, so basically it's a skull and the brain. Once we pre-process the data, the next thing that we do is de-identification. That means we also do a process which calls skull stripping. We remove the skull completely, and what we work on is just the brain. Now, believe it or not, there are out there are algorithms that if you give the skull with the brain of individuals, just the skull and the brain, basically you might feel like, okay, that's weird, but they can create the face. So that if we fail to analyze the data with the skull, even though if it is in a secure data, it's called we are creating issue with the HIPAA compliance. So our research is not HIPAA compliance. And that also consider that we are leaking PHI data, though it is in our server, though it is secure, but because we have been handling the brain with the skull, it's considered as identifier. So you need to do de-identification. So when you go dive into research, there are so many things that consider the red light when it comes to PHI. So let's see what we have next. Okay, now that you know that how to handle PHI data, now that you prove that your research is HIPAA compliance, you know how to protect your data. Now, what is it? The next thing is that in order to get approval, first of all, for your research, you have to prove that any kind of human-related research that you're doing, it doesn't cause any harm to an individual. Also, you want to show that your data is going to be secure in a secure database. You want to prove that it's HIPAA compliance. You want to prove that you're protecting PHI data. So for any research considering human being a part of it or entirely about human data, the internal review board in any organization comes to picture. So it's a group that's been approved under FDA to review your research that involves human subject. It uses a group of people to review every protocol, every material, and everything about your study that has human subject. Protocols are reviewed by IRB staff and IRB members. They pass comment, they put questions. There are two types of IRB. There is expedited. If you prove in your research study that you are not going to have any risk or the risk for the participant to participate in your study is absolutely minimal, you can take advantage of expedited IRB. So they review it and get back to you like 2 to 4 weeks. But if you are using human samples, if you're running any MRI data, if you're running PET scan, if you're using any neurofeedback or similar examples like that, or the whole MRI scan of the brain or the X-ray, which somehow you are exposing humans to some sort of chemicals or procedure, that's how it is a regular IRB, and it takes at least 6 to 8 weeks for them to review any aspects of your research to prove that it is HIPAA compliance. It's also like PHI is taken care, you're not going to harm, what are the aspects of your research. So under no circumstances, any research begins if the IRB will not approve it. So you see a lot of research going on, research studies going on. They have gone through.
One sec.
Yes. Can you mute yourself? So they have gone through all this process to make sure that individuals are not harmed, the data is protected, the identification of each individual is protected, and the studies are HIPAA compliance. So that's all about me. You might be wondering, okay, I don't collect human data. We don't do any psychological collection of data. We don't do anything about it. But again, as I started, it is the era of data. You might be at some point data scientist or being a part of a research study, or maybe one day you just go to a coffee shop, you'll see someone left a folder next to you and you open up like, oh, wow, credit card number, name, last name. So this is considered PHI. You want to find that person and pass them. You don't want to read any of those. If you are working in an organization that's doing research on human, it's just starting for you to learn about this. If you're going to be a data scientist at some point, you're going to handle some kind of human data. I hope this was informative enough to give you information about the data security and management for the human subject studies.
Thanks, Sahar. So real quick, the attendance form is going to go out in the chat window. Please fill it out for every single presentation that we're having today. Give me a second to pull it up. Wi-Fi is slow today. So Sahar talked briefly about how people can handle human data securely and ethically. And I'm going to be talking about, well, what if you don't necessarily work with human data, but you work with data of a different sort, that which is collected in a laboratory? And this might be human data, but more often than not, you're going to be collecting data on either biological specimens, physical specimens, materials that you produce, a robot that you built or that you're working with, etc. So how do we approach data collection in a laboratory setting in a way that is rigorous and ethical? And here's a question: why do we wet lab scientists need to be concerned about data integrity? Well, there's really three aspects that we're going to talk about today. And the first is intellectual property. I'll introduce the concept of what IP is and why we should be concerned about IP. We'll talk about what this means by way of integrity in research. And thirdly, we'll talk about the issue of scientific reproducibility. These things are all different concepts, but they're all related to the idea of collecting data in a way that not only puts your best foot forward as a scientist, but also ensures the integrity of the project that you're working on. Cool. If we could go to the next slide, Sahar. So how many of you have heard of the term intellectual property? Just by a show of hands. How many of you have heard of IP? Let me look in the... Okay, only 30 people have heard of IP. Here we go. Okay, some of you have found the different colored hand emojis. So intellectual property may take different shapes and forms depending on the discipline that you're working in. So for example, if you're in a computer science group, your intellectual property might exist in the form of a list of code that you've produced, or in a software that you've written, or in a particular algorithm that you've used to model some sort of system. You know, in a wet lab setting, an intellectual property entity could be something as simple as, you know, an experiment that you designed, or a cell line that you engineered, or a compound that you produced. I like the Wikipedia definition because I think it's very broad. And Wikipedia writes, intellectual property is a category of property that includes intangible creations of the human intellect. So I would say it's tangible and intangible. But anything that you intend on, or anything that has the possibility of making its way into a patent, a paper, or a marketable or commerciable entity, those things are all considered intellectual property. Now, how many of you have parents who've told you about what it's like to patent something? Anybody? We have some geeky parents here. Oh, what is that? Yep, so we got some geeky parents here. That's awesome. And, you know, maybe one of you, some of you at some point in time in your research careers will find yourselves in a position of having to talk to patent attorneys about submitting patent applications for protecting intellectual property that you and or your coworkers have produced. So why is this related to data collection? Well, in order to demonstrate ownership of intellectual property, one has to have a rigorous set of data that one has collected that indicates and demonstrates that the intellectual property is indeed yours. You know, obviously you've all heard of lawsuits that happen. Hopefully none of you have to experience any of this, but in industry, there's lots of lawsuits about who invented what first. And what do lawyers go to at the end of the day to determine who owns the intellectual property? Well, they go into people's data collection systems and see, okay, who is the first inventor of the said entity? So evidence of intellectual property, how do you demonstrate that a piece of intellectual property is indeed yours, or that you are a part of the inventorship? Three things. First of all, if you're in a wet lab setting, your laboratory notebook entries, both paper and electronic. And we'll talk about how to keep rigorous paper notebooks in a moment. And it used to be, actually, before the era of secure servers and databases, that paper notebooks were the gold standard of authentic laboratory records. And to this day, I think the statistic is 85% of research laboratories still depend heavily on paper notebooks. Raw and processed experimental data. So this means whatever data you collect in the laboratory, either on an instrument or by observation from your five senses. This stuff is, it could be qualitative data, it could be quantitative data, but this is all part of what needs to be recorded as a demonstration of production of IP. And thirdly, prototypes of novel materials, entities produced in a research endeavor. So if you're a synthetic chemist in my group, it could be a sample of a compound that you've created. Or if you're in one of the engineering groups, it could be a prototype of a robot that you made. Okay, great. We can move on to the next slide, Sahar. Okay, so I tried to distill, and those of you who are in my group heard the longer version of this, but I tried to distill a few points for keeping lab notebooks. How many of you, just by a show of hands, work in a wet lab research group here at ASDRP? How many of you work in a wet lab research group where you physically come on site to our research campus on a regular basis to do your science? Okay, so that's like a lot of you. So if you didn't know, there are laboratory notebooks that you can take in the little decontamination room in the chemistry building. There's a whole stack of unused lab notebooks, and you can take one for yourself as a means of record keeping in the laboratory. But we'll make this interactive. Here's the first point. Every experiment performed must be recorded as an entry in one's laboratory notebook, and it should be dated and time-stamped. Why is the date and time important? And maybe somebody can unmute themselves. Let's get some voices here. Anybody? Why is recording the date and time? Yeah, go ahead.
Is it because maybe, like, let's say you're testing an experiment, sometimes the amount of days that it takes for like the chemical reaction to happen would also matter, which is why you need to write down the time.
Absolutely. That's very good. So sometimes an experiment might take X number of days, and you need to record when the experiment was begun so that you know with a high degree of confidence when you make an observation later on how much time has elapsed. Why else? Why else do we date and sign and time our laboratory notebook entries?
I mean, like you said, like when they lawyers or whatever, like when they try to prove who invented something first, they're gonna go through your notebooks, right? And I guess keeping track of the date and time of when you completed it will ensure that, like, if you did make it first, you're getting that credit.
Absolutely, yeah. So to demonstrate inventorship or innovation. And I won't talk about my research career outside of ASDRP, but I had to sit through one of these situations where I had to produce, and thankfully, my notebook entries were dated in time, but I had to demonstrate from notebook entries that were created years ago that I was indeed the individual who ran those experiments at such and such a time and such a day in 2018 or whatever. So dating and timing your lab notebooks is very important. Electronic notebooks, things like RSpace, Signals lab notebooks. There's different manufacturers now for electronic lab notebooks. They create an automatic timestamp when you edit an entry. And you cannot alter the timestamp. Okay, next point. Notebook entries should be made in pen and should not be amended after the conclusion of the experiment. Why is that important? Why pen? Like, why not pencil? If you make a boo-boo, you can erase it, right? Why should you create your laboratory notebook entries in pen?
Because, like, it could be easily erased, and if, like, you spill chemical stuff on it, it's going to cover up the pencil mark because it's too light.
Okay, actually, it relates to the second point. This is a huge issue in chemistry research labs, but actually, pencil is more robust than pen in resistance to chemical exposure. Ink will degrade very quickly, and you can wash out ink. But why pen?
I think it's because you want to show that you haven't made up any data or you haven't covered up any data, because the science just should be what it is. It shouldn't be able to be changed in any way.
Precisely. Thank you. That was excellent. Okay, so in science, you have to demonstrate that the data that you have produced is rigorously well, first of all, it's rigorously objective, and also secondly, not fabricated or not amended later on after you reach some conclusion. Okay, thirdly, observations should be recorded real time by the person witnessing the observation. So what that means is you shouldn't create your lab notebook entries a day or 2 or 3 or 4 or a week after you ran the experiment. That would be an incredibly big breach of research integrity if you were to run an experiment and not create an entry in your notebook until days or weeks after the fact. And that goes with the first point. It has to be the person who is making the direct observation. So you can't text your friend and say, hey, you know, Bob, I don't have my lab notebook right now, but can you record these numbers for me? It has to be the direct observer. In fact, in some professional settings, if you miswrite something, you have to actually get somebody to co-witness the observation with you, and then they have to sign off on your notebook just to correct the number that you recorded incorrectly. Okay. In the institution that I work in, that is the standard. You cannot change anything in your lab notebook unless there's a co-witness who co-witnesses an event or a number. Fourthly, when necessary, create electronic backup copies or notebook entries and experimental notes. We've all been in situations where we lost our lab notebook, or things happen, chemical exposure, right? Notebooks might burn one day, I don't know. But it's always good to create a secure, and I mentioned secure because Rob is going to talk about this in a moment, a secure way to store data that you're collecting in the laboratory. And fifthly, if there is data outside of your notebook that corresponds with a notebook entry, for example, if you're working in a biology group and you're making observations about things under a microscope and you have images, maybe you have a fluorescence microscope time course associated with that entry, you have to indicate on the file name and in your notebook that those two things are correlated with each other, because later on when it comes time to demonstrate ownership of an experiment, that's what people are going to look at. Okay, thanks, Sahar. Maybe we can go to the next slide. So these are things that you might collect outside of a laboratory notebook: digital data, if you have spectroscopic data from an instrument, imaging data, computational data. How many of you, by show of hands, use the research server cluster in your work? How many of your groups use the cluster to run jobs? So I hope people are raising their hands if they're, yeah, okay. So quite a few of you use the server cluster for running jobs related to your research. Your computational experiments should also be recorded, if not in a physical lab notebook, in some sort of note-keeping fashion where you have entries related to, I submitted this job for this particular experiment. That's pretty standard fare in the life sciences. I would recommend when you name your spectra, and here's an incident, a little story that happened last night. I was intending to meet with a number of my research students to finish up a paper that we'd been working on for the last year. We're working on these diterpenoids that we think can modulate gene expression in cancer cells by regulating NF-kappa B activity. So that's a cool synthetic chemistry project. We made a lot of analogs of these compounds. And then yesterday, it occurred to me that the people who were working on that project hadn't really sat down to collect and to inventory all the spectroscopic data. And some of you are on this call right now. And so we ended up instead of spending that time writing the manuscript and putting together the supporting information, we spent multiple hours just inventorying NMR data, infrared data, mass spec data, collecting some of the data that was missing, and trying to figure out when people who had left the group already a year ago, somebody had named an NMR spectrum from, I don't know, October of 2020. So this is like a year ago. Named it so-and-so's long-ass 13C NMR. Like, what does that mean to the modern day scientist? Absolutely nothing. So name your files something that is understandable, not only for you in the moment, but also for somebody else later on down the road. Because we
and a bit more adult.
There we go. So please do not name things so-and-so's long ass NMR experiment. That is not only non-descriptive, but also rather unprofessional. Incidentally, that individual who took that NMR has no recollection of what that NMR was of. And so now we got a problem because we've spent all this time making compounds, collecting data that we did not assign names to and organize efficiently. And, it was a learning experience. I don't necessarily think that that was a waste of my multiple hours of my Friday evening. I think it was a good learning experience of how to rigorously inventory spectroscopic data. Okay, back up your data on a secure digital repository. Some of you know in our laboratory, in our chemistry building, we have two open access computers that are remote desktops. You can access them through TeamViewer. And one of them decided that it was going to fry the hard drive 2 weeks ago. So one of our lobby computers that students often sign into to process their data, the hard drive got completely fried. There was some sort of mechanical failure, I guess, on the computer, and the hard drive was irrecoverable. What do you think happened with all the data that was stored on that computer's hard drive and not backed up anywhere else? These things happen in research. It's gone. You're right. It's gone. Thank you, Amelia. It's long gone too. So, we make these points not to necessarily say, okay, ASDRP has crappy computers, but these things happen in research. I remember when I was an undergraduate, this happened. My laptop decided to fail, and I had not backed up a lot of the data there, and I was at a loss for words and many tears for many days because I had to resynthesize a lot of compounds. Okay, organize your data, organization. We can go to the next slide. I don't want to take up too much time. This is the last thing that we're going to talk about. Integrity and reproducibility in science. Here's a few questions, and I want this to be an open discussion. Why is it important that data is reproducible in scientific research, and what does reproducibility mean? Maybe somebody can unmute. I see people in the chat window, but maybe people can unmute themselves.
It basically means that other scientists can replicate the experiment and get the same results.
Okay, yeah. So this is actually related to the fourth question there. If science has to be reproducible, we all know of stories of so-and-so in the research group who has the magic hands, who can somehow get this experiment to work and nobody else can get the experiment to work. That's a big red flag in science, because as scientists, we want to safeguard the truth of the material that we intend on conveying. So that's why it's important to be reproducible. And we're going to talk about a story in a moment, actually a pretty crazy story in a moment, of a group that where this was a problem and how that had to be addressed. So in my group, we have what we call an open notebook policy. And what that means is that everybody's lab notebook is visible to every single other group member. We keep all of our lab notebooks on a bookshelf in the hallway in the laboratory. And, if I wanted to know, if I'm a researcher and I want to know what, I'm going to pick on one of my students. If I want to know what Udbov ran, an experiment that he ran like a month ago, I should be able to walk up to a bookshelf, open up Udbov's notebook, find an entry in there that I'm interested in, and I should be able to find a procedure that I can execute and reproduce to the point. Why do you think many labs adopt open notebook policies where everybody in the research group, obviously not people outside of the group, but everybody inside of the research group has access to everybody else's experimental records? Why is that? I think studies have shown that over 60%, over 70% of research groups have some sort of verbal policy where that is the case. Certainly in every single research group I've ever worked in, it was an open notebook policy. You had to have your notebook in the laboratory, and anybody in the group had the right to open up your notebook and query a particular entry. Any thoughts? Why is that a policy? Why does that need to be an explicit policy, and what does that do for science? And this is not just true about notebooks. This is also true about code, right? If you're a computer science students sitting here right now, you're like, okay, what does this have to do with me? You know, in many computer science groups, your code has to be visible, not only to your PI, but also to your coworkers.
Is it so people can maybe help you with your work and know what you're doing?
Okay, so on the first level, it's so people can help you and they know what you're doing. It's very important that your research advisor knows what you're doing. In the chemistry side, it's a big safety concern, right? But what else? Why else do a lot of groups have open notebook policies where everybody in the group has access to everybody else's notebook records?
It helps keep people accountable for the data and like helps
Yes.
people stay organized.
Good. And I see Gia saying this too. It's a metric. It's a way of ensuring accountability. Okay, as scientists, everything that you say that you do must be backed by an experiment, by data, by hardcore evidence. Okay, we scientists pride ourselves in the fact that nothing that we ever say or write is on a whim. It's always founded and grounded on true statements that we can verify with data. And that data has to be open for other people to interpret, to either falsify the claims that we have made or to support them. And this metric of accountability is huge, especially in modern labs where, you know, we're all into the digital age and, you know, this is my notebook, don't touch it and whatever. It makes you a better scientist to have to know when you're writing your notebook entries that, or your code, that every single other person that you will work with in that group has access to your data collection. Okay. Next slide, and then we'll close on this story, and then I'll hand it off to the CS people. Here's an interesting story, and if you're not listening up, you're not paying attention, pay attention here, because this is there's a lot of intrigue in this story. So the person on the screen there is Frances Arnold. Does anybody know why Frances Arnold is so famous? She's on the faculty at Caltech. The reason why she's famous is there's a thing on top of her picture on the screen. Anybody want to offer up why is Frances Arnold so famous? And what is she famous for? So maybe people can unmute themselves because I think the verbal communication is good.
She's famous for winning the Nobel Prize.
Exactly. And do you know what she won the Nobel Prize in? It was in 2018. I actually got to attend a seminar when she got the prize. I got to touch the prize. It was a lot of fun. But what did she win the Nobel Prize in chemistry for in 2018? Anybody know? Any of you who are biologists or who think that you're a good bioengineer or who appreciate biochemistry should know this. And if you don't already know it, you should read about it. Anybody? Directed evolution. So Frances Arnold on the faculty at Caltech won the Nobel Prize for directed evolution of enzymes using biological catalysts to do synthetic chemistry. And her group is very prolific. They published like a JAX paper or Angewandte paper, Science, Nature, Chemistry, you name it, about all the high-tier journals, like every few weeks. And she's well-deserving. She's an awesome scientist. I had the great honor of actually meeting her a few years ago when she won the Nobel Prize. Awesome scientist. So her group, two researchers in her group, Inha and Zijun, co-authored this paper in Science. And Science is the most prestigious journal in all disciplines. Okay, Science is like it's a pan-disciplinary journal, and it's got the highest impact factor of any journal, and it's probably the most prestigious academic research journal in our present day. And so they published this paper in January of 2020. This is pre-COVID, obviously. Site-selective enzymatic C amidation for the synthesis of diverse lactams, where they report the engineering, the evolution of these enzymes that can do CH amination and make these cyclic amide compounds, very important medicinal compounds. And they report that these enzymes do these at upwards of 95% enantiomeric excess with very high yields on gram scale. If we can go to Actually, I'm going to put a link in the chat window. We're going to do this as an exercise, just to keep all of you entertained. So there's going to be a link in the chat window. And this came out in Science less than a week after they published this paper. Does somebody want to read it? I'd like somebody
after publication of the report, site-selective enzymatic CH invitation for synthesis of diverse lactams, efforts to reproduce the work showed the enzymes do not catalyze the reactions with the activities and selectivities claimed. Careful examination of the first author's lab notebook then revealed missing contemporaneous entries and raw data for the key experiments. The authors are therefore retracting the paper.
Wow. And Sahara, I think I have this on the next slide. If you don't have this link open, open it up in the chat window. As a scientist, that is the most chilling thing that you can read. So what was, I don't know if all of you caught that, but in layman's terms, these authors had to retract their paper. They had to say, you know what, we goofed and we published false claims. And by the way, the first two authors on this paper were fired from her laboratory. Their research careers are more or less ruined for a single paper wherein it wasn't necessarily all false. It wasn't like they made up all the data, but their record keeping was not rigorous to the degree that one could say with confidence that these experiments were reproducible and that they were executed to the utmost of rigor. Okay, these enzymes work. I actually know somebody in the, who works in that area. These enzymes do work and they do catalyze some of those reactions. It was simply the case that, first of all, INHA's notebook did not have the entries for some of the key experiments that they published. And secondly, the numbers that were published were not in congruence with the data that was collected. Wow, that's chilling. If you read that retraction, that is one of the most chilling things that you can think about as a scientist. How two people's scientific careers were more or less destroyed by poor data keeping in the sciences. So it's chilling, but it's also, I think, encouraging because it brings to attention the importance of record keeping skills and being able to keep track of your data in a way that is, that upholds scientific integrity, that upholds scientific rigor. That's something that you can hold pride in, that you can be proud of at the end of the day and say, I have full confidence in these claims that I'm going to make. So that is, I think that was one of the most confounding scientific stories in 2020 when all of this kind of blew up. And Frances actually did a really good job of not covering up. She actually was the first person to go forward and say, we're going to retract this paper because people in my laboratory could not reproduce this individual's experiments and they could not find her lab notebook entries that correlate with this. So Frances Arnold, the principal investigator of this paper, was the first one to call for the retraction, which I thought was very noble, but also she admits openly, she did not do her job well as a principal investigator in that regard. Okay, that is all I have in terms of data keeping in the laboratory. So I think, just a quick few reminders. The data doesn't lie, and we'll talk more about this later on, I suppose, at a later session. But please do record all experimental data, whether or not it's in congruence with your initial expectations and hypotheses. Do exercise caution in coming up with conclusions based on the data. Think about statistical significance. Honestly report statistical significance. If it's not statistically significant, don't make it statistically significant just for the purposes of getting a paper. We don't do p-value massaging here at ASDRP just for the sake of publishing work that is not necessarily ready to be published. Okay, so do not massage the data to fit expectations. That will land people in a lot of trouble, and many a person have destroyed their research career doing that. Okay, cool. That's all of me talking. I think this is Rob now.
No, we're going to have to continue this because I can't possibly cover anything in 3 minutes. So let's do another session on security from here on out.
I will answer a question in the chat window. And the question is, how did the Arnold lab determine that these entries were not reproducible? I don't know exactly how that was determined to be the case, but in most research laboratories where you have an open notebook policy, anybody can walk up to your notebook and say, oh, I found this really cool. I'm going to try to reproduce this experiment. And they are in full right to do exactly that, to open up somebody's notebook and attempt to reproduce an entry that is recorded in there. And I'm sure there's a lot of people in that particular lab who were interested in the report in Science, which was published by these individuals. This is if these enzymes do what they say that they do, then this is very exciting stuff. And if I worked in that lab, my first reaction would be, gee, I want to see some of this happen too. And maybe I'll try this on my substrate. Maybe I can get their enzymes to work on my substrate. And then when things start not working, then you start looking back and say, gee, how come it worked for them and not for me? And then you try to go back and you do a little bit of archaeology in people's lab notebooks. And that can land people in trouble. Okay. So if you didn't do your attendance form yet, please fill it out. I suppose we'll get to the CS stuff at a later point in time. But any questions, either for myself, Sahar, Robert, or Suresh, who's I didn't get to talk, unfortunately.
I have a slightly more general question. When we talk about how people will go back through lab notebooks to see which research group invented something or came across something first, and how people kind of the whole idea with IP of keeping your research and your information kind of shielded from other scientists, does that ever kind of deter innovation or collaboration if people are trying to focus on keeping their IP rights?
Well, usually this integrity check happens within a research group, which is why a lot of groups have open notebook policies, including my group here at ASDRP. We have an open notebook policy. Now, it's a very good question. And two things, you should not share your lab notebooks with the outside world until the data that you intend on conveying has been protected either in a publication or in a patent. But post-publication, after submitting your work to a journal, let's say you submit your work to a journal for review, the journal editors and the journal reviewers, if your paper's undergoing peer review, they have every right to ask you to mail them your lab notebook and to look through your lab notebook and make sure that you indeed ran those experiments. They have every right to ask for your lab notebook if they're reviewing your paper. That's one of the ways in which science referees falsehood. And then secondly, once it's published, once it's in the public domain, that's open for anybody's scrutiny. And people can go out there and they can try to reproduce things. And there's many stories beyond this story that was mentioned here of people who publish stuff. And then some other research group on the other side of the world said, gee, that's really cool. And then they tried to reproduce experiments and they could not reproduce them. And that was, that blows up in people's faces and it's really ugly. Does that make sense?
Yeah, thank you.
It's a good question. Akash, you asked an excellent question. And this is, truth be told, many brilliant scientists have really terrible handwriting. Please write legibly, because if people cannot ascertain what you've written, that's very bad, because they will not be able to, with confidence, ascertain the integrity of your experiments. Okay. So there's no other questions. Hope you all learned a lot.
So Edward, I had one question.
Yeah.
So when you write dates in your notebook or write times, is there a way to verify those dates as well? Because you can write some random date and just say it happened on this day.
Yeah, science relies on the integrity of the scientist. So it's kind of like a honor code. Yeah. And we scientists take that honor code very seriously. You know, one breach of that honor code and your career is over. So, you know, I think as scientists, people hold themselves to a very high degree of integrity in that regard. Yeah, obviously with an electronic notebook, the computer records the timestamp for you, so you can't change it even if you wanted to.
One small thing you can do is ensure that the timestamps are written in chronological order. That too.
Yeah, when people have notebook entries that are not chronological, it, you know, is it's glaring.
It's at least an orange flag, if not red.
Yes.
There have been cases of lab notebooks being examined using chromatography to find out if the inks used were contemporaneous with the time at which they were supposedly written. They did that with Tesla's notebooks and with Edison's, I think. So they'll go that far if they have to.
Okay, yeah, if you weren't chilled by the Francis Arnold thing, read it, because that is a very chilling thing to hear as a scientist. That's a good question that Varsha asked. What if there's an error in your notebook? Typically, the standard practice in most research labs is you cross it out with a single strike through, so people can still read what you had written incorrectly, and then you have a witness sign next to the strikeout. So your lab partner or somebody else who saw the data for what it was and knows that with confidence that you incorrectly recorded a correct point of data. And they would then sign off saying, I witnessed this. I witnessed this strike through.